In this video, we will discuss about another new technique, that is deterministic barcoding in tissue, and it is popularly known as DBiT technique. So this technique has been used to identify the spatial description of the genes that are present within the tissue, or in the later it can be used to identify the genes that are disrupted because of their extracellular matrix, or because of their because of their surroundings. And that is usually occurs whenever the cells differentiate from one phenotype to another phenotype. The surrounding factors will also surrounding phenotypes will also play a role. So instead of using just the single genes in a in vitro experiment, it is used to it is beneficial to identify the genes directly from the tissue. So, and before this, there are several other techniques has also been developed, but those techniques can identify only a few hundreds of genes, whereas this techniques can identify uh, thousands of genes at a better accuracy. So let us go through their sample preparation workflow. So first they have take this glass slide with the glass with a tissue slide, and then add this and antibody derived DNA tags. And thereafter, in this first step, uh, they have used this barcode labels, oligo DNase tags that can identify the mRNA that are present within the tissue. And also they did another experiment where they have used these antibody tags to detect the protein of interest, and they have used the micro channel plates of different size. So what happens in the first case of the experiment is that this whenever this oligo DNA text detect the mRNA, it will trigger this reverse transcription, and that will lead to these cDNA strips that are available within those cells. And the poly A tails will also present along with the mRNA. And in the other case, the protein antibody derived DNA tags can bind to the protein of interest along with this poly A tails. In the second step, they use this second PDMS macrochannel plate in a perpendicular directions. So in this case, only the barcodes will attach to that locations where these poly A tails are present, uh, or where these poly A tails are present, that is either at the mRNA with the poly A tails, or this protein of interest at the poly A tails. So from combining both the cases, we can now identify the XY positions of the cells based on the two barcodes that are present, that is barcodes through this A1 A2 and the barcodes through B1 B2. So this is the first step, and this is the second step. After second step, how it will looks like, that is the proteins that is attached after the barcode 2. This is the this you can consider it as the barcode 2 green one, and the this one is the barcode 1. So now this is what it looks like after this first and second step. And now it can, if these tags are fluorescence level, then it can be measured in the microscope, and or they can prepare the NGS samples and do the NGS sequencing, and thereafter they can based on the barcode locations, they can identify which kind of mRNA or proteins are present at which position of the tissue. And from this DBiT techniques, they can able to identify more than 22,500 genes, and they have used this experiment for around 20. They said that they can use this technique for around 2200 proteins detection. And in the for examples, in order to show an example, they have used 22 protein channels. And this is how the microtubule plates at the first and second step looks like. So in order to avoid leakage, they provide the clamping of this microtubule plates of the smaller size. And this is just an example with fluorescence level barcodes, that what they have done. So in the first step, they use this Psi3 fluorescence label barcode in the one directions, and the second step they have used this FITC labels barcode in the orthogonal directions. And since from the requirement, they have the second flow, or the FITC label barcodes will only bind to a place where there is red Psi3 label barcodes are available. So because of this, the FITC label barcodes will be shown in the square boxes. and that we can see in this FITC fluorescence channel, that FITC labeled barcodes are in the square boxes. After validating these results, they need to check whether these models, how accurate are these models in the actual tissue experiments. So for this they use the endothelial cells and they grow the monolayer of endothelial cells, which can be a better model, realistic model of the tissue. And thereafter they label the junction of the endothelial cells and also the DAPI. And from this study, they can see that even though the channel is cutting the cells at the intermediate, they do not see any leakage of the channel. For example, in this case, we can see the DAPI only at the 1 pixel, and its DAPI signal is not leaking into the other channel, or not leaking into the other pixels. And these are the zoom in views, and we can see the DAPI signals are not leaking into the other channels, and for this case also, and for this case also. So this indicates that their DBiT techniques are has DBiT techniques, in the DBiT techniques there is a less probability of the signals to get leaks from one pixels to another pixels. And after that they check this image. They check this DBiT technique at the different channel length, macro channel width. So at one at different macro channel width, they can also see that the fluorescence labels, fluorescence barcode A will not leak from one channels to the another channels. And when they did the actual tissue imaging using DBiT, and they can see that in that also that the FITS channels are just in the square box, and there are not leakage, there is no leakage of the one channel into the another channel, and that can be seen in this panel F. And after that they wanted to compare their they wanted to quantify those leakage that they have observed. So when it comes to the diffusion distance, they can see that for the 10 micrometer channel, the diffusion of the signals are around 0.9 plus minus 0.1. It is within the 10%. And same things they have observed for 50 micrometer channel, that is around 4.5 micrometer plus minus 1 micrometers. So this is also within the 1%. And also one thing that they mentioned, that these deviations are maximums that are present at the edge of this microtubule channel plates. So these are the maximum deviations. However, within the channel, however within this macrotubule or this macro channel plates, within this macro channel plates, not tubule, within these macro channel plates, the deviations are much less. And when it's come to the pixel size, how much pixel size they can see in the image and how much they are different from the theoretical pixel size, and they can see the 10 micrometer pixel size, they can see about 9.1 plus minus 0.3, and for 50 micrometer channel they can see the 52.5 plus minus 1.3. So they are also close to the theoretical values. And thereafter they check how much cells are present within the 1 pixels. For 10 micrometer channels, they can see that around 1.7 cells are present, plus minus 0.4, and for the 50 micrometer channels there are around 25 plus minus 8.8. So this gives the insight that if someone needs to measure only the single cells information, then they can go for 10 micrometer macro channel plate. And if they are okay with this multiple channels, multiple cells of similar characteristics, multiple cells in the same pixels, then they can use this 50 micrometer channels. And next they compare their study using they compare their method with the previously available method. And they can see that for spatial transcriptomes, it can give the number of genes around 7500, which is similar to the DBiT sequence of 50 micrometer micro micro channels plate. So this indicates that the DBiT is more sensitive. It can detect almost same number of genes and at different microchannel length, at smaller microchannel lengths. And same thing, it works out. Same things has been seen for Visium method. That is, it can detects around 5000 proteins at 55 micrometer channel, and which is equivalent to the are almost similar, not equivalent, almost similar to this DBiT, which can detect similar number of proteins at around 10 micrometer channel width. So this indicates that DBiT are more efficient and sensitive compared to the previous case. For this slide 6, it can detect very less number of genes compared to both the DBiT and other methods that are available. Next, they check what are the unique molecular identifiers that different techniques can identify, and they can see that for this DBiT, and for both the microchannels that they have compared, microchannel width that they have compared, it can detect more number of UMI or unique molecular identifier compared to this special transcriptome or visium, and also compared to this slide C. So this figure so far points out gives us the outcome that DBiT is much more better than the previously established techniques, and it can be used more. It can be used in a more sophisticated manner to not just to identify special transcriptomes in the normal tissues, but also in the cancer tissues where migrations are pretty much been affected by the surrounding environment, such as